Hello, Mr. Allman here, and this is your Allmencast on gel electrophoresis. Electrophoresis is a process that uses the physical and chemical properties in order to analyze DNA or proteins. The advantage that's taken here is that DNA has a negative charge. The phosphate groups on the backbone of the DNA molecule have oxygen atoms that are ions, that are negatively charged. So if you expose DNA to an electric current, the negatively charged DNA molecule will move towards a positive electrode. The way that that's done is to be able to first create a gel. A gel is going to be a suspension of water molecules in a matrix of another molecule. Jello is an example of a gel that's made using the protein collagen, which comes from animals. In this case, you're going to be using agar, which is a common carbohydrate that can be formed into a gel. The first step in the lab is to be able to measure out and take agarose, which is that carbohydrate powder, and combine this with some buffer solution, heat it up, and dissolve the agarose in that heated liquid mixture. It takes that heat to be able to separate the agarose molecules from each other so that they dissolve and form a solution. Once you've got that near boiling and fully dissolved, you're then going to pour the cooled solution into an electrophoresis tray. The tray consists of a small plastic piece which you're going to put the rubber dams on either side to create a space into which you can pour your solution. So once that solution has cooled, you pour that into the chamber, remembering to add in the comb. The comb will fit onto one side of the gel. The liquid is going to surround the bottom parts of that plastic, and then once the gel is cooled, you'll be able to pull that out. And when you pull that out, there's going to be indentations in the gel, which are called wells. And those wells are what you're going to put your DNA sample into. Once you have allowed that to cool, you can then take out the comb, take off the dams from either side, and then you can add your gel into the chamber. This is the electrophoresis chamber. There's a little notch into which your tray will be able to sit, and now you're going to have a gel that is trapped here in a chamber that is going to hold liquid. The liquid that you're going to fill that with is electrophoresis buffer. It's essentially a very fancy salt water, and because of the ions that are in this buffer, the liquid will be able to conduct electricity. The way that you move your DNA samples is using this. This is called a micropipette. You'll notice on the top, it's going to list a number of microliters, and that's the amount of volume that this device is capable of being able to move. The dial on the side gives you a reading for exactly how many microliters are being moved at a time. By turning the knob at the top, you can adjust to increase or decrease the amount of liquid that's going to be moved. Because DNA is a fairly small molecule, even after multiplying it many times, we usually analyze very, very minute amounts of DNA. So we need a special device in order to be able to actually measure that out. The way that this works is that you take a micropipette tip, which is a small translucent plastic tip, you put that onto the end of the micropipette. In order to operate it, you push it down, you put it into the liquid that you're trying to move, for example, your DNA sample. When the tip is submerged, you lift up, and that is going to draw the liquid up into the tip. Then you would move over to the chamber, and with a steady hand, you're going to put the tip into the well, being careful not to break it, and push down all the way on the micropipette in order to squirt that small DNA sample into the well. You then pull out the micropipette, and once it's out, only then should you release the pressure on the trigger. Otherwise, you're going to end up drawing the DNA back up into the tip on your way out. you would repeat that process, adding in a different DNA sample into each well. When you add the DNA into those wells, you want to leave the wells on the outside empty, because you're going to get a more consistent result if you use only the wells that are in the center of the gel. With the DNA in there, and with the DNA surrounded and the gel surrounded by electrophoresis buffer, you're then going to put on the lid. Now, there are 2 wires attached to that lid. The color of the wires is there as a help, but the color of the wire is actually not significant. What is important is where, on your power source, you plug those wires into. Now, because DNA is negatively charged, and I put my DNA sample on the right side of this chamber, I know that that negatively charged DNA needs to move towards the positively charged electrode. So this red wire needs to be connected to the positive terminal on my power source. I would want this end then to be plugged into the negative side of my power source. Once the wires are connected and I've double and triple checked that I've set up my positive and negative electrodes properly, I can then turn on my power source, and I would recommend that you use 150 volts, because the higher the voltage, the faster your DNA is going to move. So that's going to help you to be able to get the DNA to move far enough in a class period so that you can take care of the analysis. Once you've looked in your lab protocol and you've seen the appropriate amount of time has passed, then you can turn that off and disconnect the wires. Now, there is a little bit of blue stain that's added to the DNA sample that you add into the wells. Now, the blue there is not actually DNA. That is an additional molecule that has a color to it that's simply added there to give you a kind of hint as to where the DNA is located. The DNA itself doesn't have any color. So after this process is over, you need to take off the lid, remove the gel, put that gel into one of these white trays, and then you need to add stain to it. And you need to add a stain that's going to attach to the DNA molecules. And the one that I'm going to recommend is the InstaStain Blue cards. So the blue here, the blue color, is a molecule that is going to be attracted to the DNA that's trapped inside the gel. By adding this to the surface, it's going to cause the whole gel to turn blue. You then need to go through a de-staining process to remove some of the excess blue color that's there. So the entire gel will still be blue, but the part where the DNA is located should appear darker. So you should see a darker band of blue within that gel. In order to help you to be able to see that, using a white light background to illuminate your gel is going to allow you to look and to find the DNA a little bit more easily. Now, going back to the concept here, you have to imagine this gel as a kind of molecular net. All of these molecules of agar are overlapping, and there's just very small spaces through which the DNA molecules can move. The DNA wants to move through the gel because it wants to go towards that positively charged electrode. So big pieces of DNA are going to get caught up in that net. They're not going to be able to move through. They're going to have to wiggle around, find those little spaces through which that they can move. But small pieces of DNA, they're going to be able to fit through that net much more quickly. And so, because the time is constant, little pieces of DNA should migrate further along the length of the gel, whereas big pieces of DNA will travel a shorter distance. So analysis of DNA, we can qualitatively make comparisons from one lane to the next and compare DNA sample 1 to DNA sample 2. But also, if you add in a marker that has DNA of known lengths, you can actually quantify and determine the exact size of a piece of DNA that has run through a gel. Now, there are lots of variables that could be adjusted here. You can adjust the amount of agar that has been added, what the concentration of agar is in the gel. That makes the net either more loose or more tight. You could adjust the voltage of your power source. You could adjust the size of the DNA that is being added in. Additionally, just to go quickly into some of the applications here, we can use electrophoresis as a way to be able to identify suspects in a crime. So fingerprint analysis or paternity and maternity tests often use gel electrophoresis as a way to be able to identify a suspect. Now, as one final piece here, in order for electrophoresis to be effective, in most cases, we need to take DNA and chop it into smaller pieces. So a special type of enzyme called a restriction enzyme is used to be able to cut the DNA. Restriction enzymes are taken actually from bacteria, and they're a defense mechanism used by bacteria as a way to fight against viral DNA that might be trying to attack them. So those restriction enzymes should be added to the DNA to cut the DNA into smaller pieces before the DNA sample is added into the gel. That way, instead of one big blob of DNA all moving together, you're going to see several smaller bands representing little bits of that DNA after it has been cut. And because every piece of DNA from a different person could be unique, those enzymes will cut the DNA at different spots, leading to a different pattern that you can analyze then on the gel. This same process can be applied to proteins. However, a protein, in order for it to be analyzed, it must have a net electric charge. Some proteins are charged and some are uncharged. So hemoglobin, because it does have an electric charge, you could analyze hemoglobin with an electrophoresis process, but other proteins, this would not be effective. As always, if you have any questions, please let me know. Thank you. Good luck.